So, if you are 60 years old right now, your immune system might secretly be 80.
Yeah, which is a wild thing to think about.
Right. And the terrifying part is that until recently, scientists couldn't definitively tell you why your body was aging faster than, the calendar on your wall. I mean, we see it all the time. Two people hit their 70th birthday. One is like signing up for a half marathon, and the other is winded just walking from the kitchen to the living room.
Exactly.
And today, we actually have the answer to why that happens.
We do. And it is a massive breakthrough. I mean, we are finally moving past this idea that aging is just some mystical, inevitable ticking clock. The research we are looking at today maps out the exact biological machinery that drives the breakdown of our bodies.
Okay, let's unpack this. We are diving into a 2026 paper from Cell Genomics by Liu and their team. And the title is Get Ready for This Interferon-Related Inflammaging Links Epigenetic Age Acceleration to Multimorbidity.
A mouthful.
It's a very dense title. It is packed with data from multiple cohorts. But the mission for this deep dive is actually pretty clear. We are going to decipher how chronic inflammation is actively rewriting our DNA to make us age faster. And, probably more importantly, how scientists are figuring out how to pause that process.
Right. So to understand how the body accelerates its own aging, we first need to look at what's happening to our primary defense mechanism over time. The paper highlights this dual threat that emerges in an aging immune system. And those two threats are inflammaging and immunosenescence.
Okay, those sound like two sides of a very uncomfortable coin. I mean, inflammaging sounds fairly self-explanatory, right? Inflammation tied to aging. But what is the actual mechanism there?
So think of inflammaging as a state of chronic low-grade systemic inflammation. Over decades, due to external environmental factors and, internal cellular damage, your body just starts producing inflammatory signals constantly. And this happens even when there is absolutely no infection to fight. Your immune system is just simmering, basically releasing chemicals into your bloodstream 24/7.
Just constantly on edge. And then the second half of that threat, immunosenescence.
Right, so that is the defective responsiveness of the immune system when a genuine threat finally does arrive. Because the system has been simmering unnecessarily for so long, when a real virus or bacteria enters the body, your immune cells are sluggish.
Because they're exhausted.
Exactly. They are totally exhausted. They fail to mount a coordinated, effective defense.
I was trying to visualize how these two concepts interact, and I keep coming back to like a home security system.
Oh, I like that.
Yeah, imagine your immune system is wired into your house. Inflammaging is when the system starts glitching. So the siren is constantly blaring at a low volume for absolutely no reason. And it's driving you crazy, it's draining the battery pack, and the constant vibration is literally causing structural damage to the drywall over time.
That is actually a highly accurate way to picture it, that chronic inflammation is essentially friendly fire. The body's attacking its own tissues because the alarm just won't shut off.
Right. And then immunosenescence is when a burglar actually breaks a window and climbs into the house. But because the alarm system is so drained and glitchy from blaring all year, it completely fails to notice the intruder. The system doesn't trigger when you actually need it to save you.
Yeah. And this dual failure has devastating clinical consequences. The constant wear and tear of inflammaging predisposes individuals to a massive spectrum of issues. We're talking cardiovascular disease, neurodegenerative conditions, and widespread inflammatory diseases.
It's just a cascading failure.
Pretty much. Your body spends all its energy on false alarms, leaving it defenseless against real threats and causing collateral damage to your organs along the way.
Okay, so to crystallize this first crucial concept for you listening, getting older triggers a double-edged sword in our biology. First, we develop this constant low-grade internal fire, which is inflammaging. Second, that fire directly leads to a sluggish, defective response to real microbial invaders, which is immunosenescence.
Exactly.
So think about your own body for a second. Have you noticed it taking slightly longer to bounce back from a common cold now compared to, say, a decade ago? That is immunosenescence at work. Your internal security system is failing to catch the burglar in time.
This raises a really important question, though. We can observe that a 70-year-old takes longer to heal. But how are these researchers mathematically proving that one person's immune system is biologically older than another's? I mean, you can't just draw blood, look at it under a microscope, and read a born-on date.
Right, which takes us directly into the mechanism of epigenetic clocks. And I was completely fascinated by how they quantify this biological wear and tear. They use a process called DNA methylation.
Yeah, DNA methylation is key here. To truly grasp it, we have to look at the architecture of our genetics. You have your DNA sequence, right? The hard-coded blueprint you are born with. But layered on top of that code is the epigenome. DNA methylation involves these tiny chemical tags called methyl groups that physically attach to specific regions of your DNA.
And they don't change the underlying genetic code, do they? They just change how the body reads it.
Exactly. They act like microscopic physical barricades. When a methyl group attaches to a gene, it physically blocks the cellular machinery from reading that part of the DNA. It effectively turns the gene off.
It's like looking at a grand piano. The DNA represents the keys, which never change. You are born with those exact keys. But DNA methylation is like someone coming along and taping down a dozen specific keys so they can no longer be played.
That's a great visual.
Right, the instrument is exactly the same, but the song it produces is entirely different.
And what's fascinating here is that the pattern of these taped down keys, the pattern of DNA methylation, changes in highly predictable ways as we age. It is not random. By sequencing these methylation patterns in your blood cells, scientists can run the data through an algorithm and calculate your exact epigenetic age, or epi-age.
Which introduces the core metric of this entire study, epigenetic age acceleration, or EAA. And EAA is just the mathematical gap between the calendar on your wall and the biological age of your cells.
Right.
If you are 60 chronologically, but your methylation patterns match a 70-year-old, you have positive EAA. You are aging biologically faster than time is passing.
Now, the researchers didn't just rely on one generic algorithm to measure this. They actually utilized 4 distinct epigenetic clocks. And they fall into 2 very different categories.
Okay, let's break those down.
The first category consists of the lifespan clocks, specifically the Horvath and Hannum clocks. These algorithms were trained primarily to predict chronological age and basically standard time to death.
But the researchers found those weren't actually the most useful for tracking disease, right?
Yeah, correct. The second category is where the real insight lies, and those are the health span clocks, named GrimAge and PhenoAge.
GrimAge is such an intense name.
It really is. But these clocks were trained differently. Instead of just looking at time passing, they were designed to predict physiological health, the onset of clinical disease, and your overall health span. They predict how many healthy, functional years you actually have left.
Here's where it gets really interesting. To test the accuracy of these clocks, the researchers analyzed something called the BCG prime cohort. This was a specific group of 384 elderly individuals. And the defining characteristic of this group was that every single person had comorbidities. Actually, many of them suffered from multimorbidity.
And multimorbidity is a critical concept in aging research. It means suffering from multiple chronic diseases across completely different physiological systems simultaneously. So we're talking about a patient managing cardiovascular disease, early-stage cancer, and metabolic dysfunction all at once. It represents systemic biological collapse.
Wow. And when the researchers cross-referenced the methylation data with the clinical health of that BCG prime cohort, the lifespan clocks, Horvath and Hannum, they did not show a strong, reliable link to those diseases.
No, they didn't.
But the health span clocks, especially GrimAge, mapped almost perfectly to frailty and multimorbidity. GrimAge isn't just counting days. It is acting like the ultimate check engine light for your body's systems. If your GrimAge is accelerating, your internal engine is failing.
Yeah, it proves that these specific methylation patterns are a direct reflection of physiological damage, not just the passage of time.
Let's do a quick synthesis of how we measure this. First, we are tracking biological aging through epigenetic age acceleration. Second, we measure that acceleration by looking at DNA methylation, the chemical tags acting like tape on our genetic piano keys. And third, the data shows that health span algorithms like GrimAge and PhenoAge are incredibly accurate at predicting real-world physical frailty and multimorbidity.
Which forces a very personal realization, I think.
It really does. So to you listening right now, imagine logging into a medical portal tomorrow morning and seeing a dashboard with your epigenetic clocks. Which number would you be more terrified to look at? The one predicting the exact year your lifespan ends, or your health span clock showing you exactly how many vibrant, mobile, disease-free years you actually have left before the biological breakdown truly takes over?
It is a sobering thought experiment, for sure. But it leads us to the most vital and arguably the most difficult part of the research. We have established that epigenetic age acceleration is heavily linked to severe disease. We also know that chronic inflammation, or inflammaging, is happening at the exact same time. But establishing a link is not the same as establishing a cause.
Wait, hold on. Let me play devil's advocate. I'm looking at this and thinking, gray hair and lower back pain happen at the exact same time in elderly populations.
Huh.
But my gray hair isn't causing my spine to ache.
Right.
Aren't they just seeing two things, inflammation and methylation changes happening to older people simultaneously? How did they know the inflammation is actually driving the aging?
You've just identified the biggest trap in observational science, confusing correlation with causation. To solve this, the researchers couldn't just look at standard blood tests. They had to utilize a highly sophisticated analytical framework called Mendelian randomization.
Okay.
And it sits near the very top of the hierarchy of evidence for proving causality.
Mendelian randomization. Let's break that down because it sounds like something straight out of a theoretical physics textbook. How does it actually separate correlation from causation?
It operates on a brilliant premise.
Yeah.
In a normal observational study, if you see high inflammation and fast aging, you have massive lifestyle confounders.
Right.
Maybe the people with high inflammation also smoke a pack a day, eat highly processed diets, and never exercise. You cannot untangle their lifestyle from their biology.
Right. The lifestyle noise completely drowns out the biological signal.
Exactly. Mendelian randomization bypasses the lifestyle noise entirely by using your genetics as an instrument. When you are conceived, you randomly inherit specific genetic variants. Some of these variants naturally dictate the baseline levels of certain proteins your body will produce over your entire lifetime.
Oh, I see.
Yeah, this genetic assignment is totally random, and crucially, it happens before you ever have the chance to smoke a cigarette or eat a single piece of junk food.
So MEMAR acts like nature's own randomized clinical trial.
Precisely. By analyzing massive genetic databases, researchers can isolate the individuals whose inherited genes naturally predispose them to produce higher baseline levels of specific inflammatory proteins. If those specific individuals consistently show accelerated epigenetic aging, you've proven a causal biological link.
Because a poor diet cannot change the genetic variants you were born with. The genes cause the protein levels, and the protein levels cause the aging.
Exactly.
Okay, that is an incredibly elegant way to filter out the noise. They used our inherited DNA to prove the exact sequence of events. And when they ran this Mendelian randomization, they caught the actual molecular culprits red-handed. They identified 4 specific inflammatory cytokines circulating in the blood that actively drive epigenetic aging.
Yeah, high blood concentrations of CXCL9, CXCL10, CCL11, and IL-18.
That's CXCL9, CCL11, and IL-18.
Right. The Mendelian randomization proved these are not just innocent bystanders floating around while your body gets older. These 4 specific proteins are the causal drivers pushing your GrimAge and PhenoAge clocks forward.
But what ties those 4 together? Why CXCL9, 10, CCL11, and IL-18 specifically?
Well, what's fascinating here is, if we connect this to the bigger picture, all 4 of these cytokines are deeply integrated into a specific biological highway, and that's the interferon signaling pathway.
Okay.
Interferons are normally crucial signaling proteins. Your host cells make and release them primarily in response to viral infections. They're the initial flare gun of the immune system.
But wait, if they are the flare gun designed to save us from viruses, why are they the exact things driving the acceleration of our biological clocks?
Because of the dosage and the duration. Go back to our home security analogy. A flare gun is great if you use it once to signal for help. But if you are firing a flare gun inside your living room every hour for 20 years, you are going to burn the house down. Chronic activation of the interferon pathway over decades leads to a catastrophic biological consequence known as stem cell exhaustion.
Let's look closely at that mechanism because it's vital. Stem cells are the foundational factories of the body, right? Like in the bone marrow, hematopoietic stem cells are responsible for constantly churning out our fresh blood and immune cells.
Right. And when the interferon alarm is blaring 2047, those stem cell factories are forced into overdrive. They are frantically churning out immune cells to fight a battle that doesn't actually exist. Eventually, the factories burn out. The stem cells lose their ability to divide and function.
So the system literally runs out of replacement parts.
Exactly. This exhaustion is a primary hallmark of advanced senescence.
And the data backing this up is incredibly robust. The researchers conducted a sex-stratified analysis. They looked at 243 males and 141 females separately, just to ensure this interferon-driven aging wasn't just, a quirk of one gender's hormonal makeup. And the causality held up perfectly across the board.
It's a universal human mechanism. Inflammation from the interferon pathway exhausts the stem cells, which then triggers the specific DNA methylation patterns that algorithms like GrimAge detect as accelerated aging.
So let's lock in this massive revelation. First, Mendelian randomization allows scientists to bypass lifestyle noise and prove causality using genetic inheritance. Second, through this, they proved that 4 specific interferon-related proteins actively drive our biological aging. They keep the immune system in a state of chronic panic until our stem cells are entirely exhausted.
Yeah, that's exactly it.
So think about the implications of that for your daily routine. Knowing that specific inflammatory proteins are hitting the fast-forward button on your biological age, what hidden triggers might be lurking in your life? Is it chronic psychological stress, a poor sleep schedule, an untreated microinfection? Every time that interferon pathway flares up unnecessarily, your cells are taping down another piano key.
Which brings us to the ultimate functional test. We have this biologically aged, stem cell exhausted immune system. What happens when a real pathogen finally arrives?
That is the fourth and final piece of this puzzle, the pathogen stress test. The researchers basically put these aging immune cells into a biological fighting ring to see exactly how they would fail.
They did. They pulled data from the BCG prime cohort we discussed earlier, alongside another cohort called 500FG, which included 212 individuals. From these participants, they extracted PBMCs.
Peripheral blood mononuclear cells. So these are the frontline soldiers circulating in your bloodstream, right? Your P cells, your monocytes.
Exactly. They cultured these immune cells in a Petri dish and subjected them to an exhaustive, highly calculated list of actual pathogens. They wanted to measure the functional output of a biologically aged cell versus a biologically young cell.
And they didn't just use one type of germ. They threw an entire rogues gallery at these cells to see if the failure was specific or systemic. I mean, they hit them with bacterial threats like staph infections, E. coli, and the bacteria that causes Lyme disease. They hit them with fungal threats like Candida and Cryptococcus.
And they also heavily tested viral responses. They utilized influenza and multiple strains of SARS-CoV-2.
Like the original Wuhan strain, Delta, Omicron.
Right. And to ensure they were testing the fundamental sensory mechanisms of the immune cells, they used TLR ligands, like LPS and RA48. These are synthetic molecules that mimic the structures of bacteria and viruses to trick the cells into thinking they were under attack.
So it was a comprehensive stress test across every single category of invader. And the results perfectly illustrate why aging is so dangerous. Accelerated epigenetic age was strongly associated with massive cytokine dysregulation. When these aged cells were attacked, they didn't just mount a uniformly weak response, they panicked.
The response is entirely chaotic. Depending on the pathogen, the epigenetically older cells would massively overreact, dumping the wrong types of inflammatory cytokines that would essentially just damage surrounding tissue.
Just blind firing.
Exactly. And simultaneously, they would severely underreact by failing to produce the specific targeted cytokines needed to actually neutralize the virus or bacteria in front of them.
It's the immunosenescence we talked about at the very beginning. The security guard isn't just asleep. He wakes up, shoots the television, and lets the burglar walk right out the front door.
That's a perfect way to describe it.
This chaotic dysregulation is the exact reason why a virus like COVID-19 or a standard seasonal flu can be easily brushed off by a 20-year-old, but can trigger a catastrophic systemic cascade in a 75-year-old. The cellular factories are exhausted, the DNA is barricaded by methyl groups, and the defense strategy is just broken.
But, this shouldn't be viewed purely as a grim reality check. In the realm of biomedical science, identifying the exact mechanism of failure is the first and most difficult step toward a cure.
Right.
Because this paper proves that the interferon pathway is the causal driver of epigenetic aging, it gives the pharmaceutical and medical communities a highly specific target.
To quickly summarize this final section, when exposed to real pathogens, from COVID-19 to staph bacteria, older, epigenetically accelerated immune cells show severe dysregulation in cytokine production. They overreact and underreact at the worst possible times. So I have to ask you, if we stress tested your immune cells today against that massive list of pathogens, how well regulated do you think their response would be?
It's a scary thought.
So what does this all mean for the future of human health? Let's synthesize the journey we've just taken. We started by questioning why our biological age diverges so wildly from our chronological age. The answer lies in the accumulation of chemical barricades on our DNA, measured by health span algorithms like GrimAge.
Right.
But this DNA methylation isn't random. Using Mendelian randomization, scientists proved it's causally driven by chronic inflammation, specifically the interferon signaling pathway. Constant exposure to proteins like CXCL9 and IL-18 exhausts our stem cells, ages our DNA, and results in a chaotic, broken immune response when we finally face a real pathogen.
The ultimate takeaway here is a monumental paradigm shift in how we view human biology. Aging is no longer a mysterious inevitability that we just have to quietly accept.
Yeah.
It is a highly specific, measurable, and mechanically driven biochemical pathway. And because we now know the pathway, we can build the roadblocks. Future medical interventions, whether they're targeted antiviral therapeutics, specific dietary protocols, or engineered drugs that suppress runaway interferon, signaling they could fundamentally slow down or perhaps even reverse the molecular aging process.
Which leaves us with a truly provocative reality to consider. Imagine we are sitting here 10 years from now. Science takes the mechanistic blueprint from this exact paper and develops a targeted daily therapeutic that safely regulates your interferon pathway. A pill that effectively pauses your GrimAge clock, treating biological aging not as a tragic fact of life, but as a curable inflammatory disease.
It's entirely possible.
If you knew you could maintain the cellular resilience and immune regulation of a 40-year-old well into your 80s, how would that fundamentally change the way you plan the rest of your life? If you went into a clinic today and they threw your immune cells into a petri dish with COVID and staph, you might be terrified of the chaos they'd find. But tomorrow, that chaos might just be a curable condition. Thank you for joining us on this deep dive. Keep questioning the biological rules you've been told are permanent, and we will see you next time.